So you just got your data back from the sequencer, and you have this really big FastQ file, and you're wondering, what do I do now? Well, I suggest that the first thing you do is look at your post-sequencing quality and make sure that your libraries look how you expect them to. This is the file that you get back from the sequencer. It's called a FastQ file, and each 4 lines in this file represent a single read. I know it looks like gibberish, but there's actually a lot of information here. The first line is your specific read identifier. This line tells you where this was sequenced, what the lane of sequencing was, where on the specific lane that this sequence was found, as well as which read it was the first read or the second read, and the specific barcode you used for this library. The second line is the specific sequence, or the string of nucleotides that was read by the sequencer. The length of this line will depend on your length of sequencing. In this case, it was a 50 base pair reads, but it could be 100 if it was a 100 base pair read. The third line in this case is just a plus, which is a placeholder. However, in some FastQ files, it will just be a repeat of your read identifier line. And the fourth line here represents your quality. It's going to be the same length as your sequence line because each letter represents the quality at that base pair. This could be a letter, number, or a symbol, and this is an ASCII character that represents the quality score as determined by the sequencer. So first I want to say you can open up this file and look at it, but never change it because this is important information about the actual sequencing. And the next thing I want to say is don't be overwhelmed by this. I can't look at this and tell you what everything is. But what I can do is put it into a program that can give me information about my sequencing as a whole, and that's what we're going to do. This program we're going to use can identify several sources of library read quality problems, and it has basically, I've broken them into 2 different sections, the sequencer problems and then library problems it can identify. So first, sequencer problems is just read quality, which aggregates the information from the quality scores into some helpful reports. The next is to look at library problems, and you can see problems in your libraries through %GC content. You can also determine the complexity, you can also somewhat determine the complexity of your library, as well as any adapter or primer contamination, and then also biological contamination, such as ribosomal RNA or overrepresented genes. We're going to be using this program called FastQC. It produces a high throughput sequencing quality control report. In a later video, I'm going to show you how to run this program. In this video, I'm just going to show you what the report looks like and what we can learn about our libraries by looking at these reports. There is also information on the website about where to go to download this for yourself. And in the next video, I'm actually going to be showing you how to run it on our cluster, but you can run it on your own computer if you want. So the first thing that FastQC does is it produces this report, and it's an HTML file, and this is what the top of the report looks like. It tells you when you ran it and what the file name was, which is great for record keeping. But it also gives you information here about your total number of sequences that are flagged for poor quality, average GC content, and then on the left is the summary, and this is all the different reports you can get from the FastQC. Like I showed you, this is the basic statistics, but there are a lot of other reports that FastQC produces that can tell you a little bit about your library. And next to these reports, it has this sort of stoplight. Green is go and red is stop. I would not live or die by these warnings, but it will tell you where in your library there may be problems. So let's get into what some of these are. We want to assess the sequencing quality. So FastQC assesses the quality of the sequences in 3 different ways. First, it looks at the per base sequencing quality. And what the per base sequencing quality is the average quality at each base pair. And so this is looking at all of your reads as an aggregate. Across the x-axis here, you have the position in the read, whether it's read 1, position 1 or position 51 at the end of the read. And on the y-axis, you have the quality score. Now this is a really good read. You have high quality, all in the green zone. What you can also see from this is as the read continues, the quality actually drops. And this is common for Illumina sequencers, and especially the older ones. The newer ones have a much lower drop in this quality length. Here I'm showing an example where you might be concerned about your reads. In this case, there was a mid-read drop in quality, and this may affect some downstream mapping efficiency. So here, right about 35 base pairs, you see a drop in quality, at least for some of the reads, because like I mentioned, this is an average of all of your reads, so a portion of the reads have a severe drop in quality. You could filter out these reads that have low quality using a trimmer, which we will talk about at a later time. If this was a really bad drop and almost all of your reads had a drop in sequencing quality, I would consider going back to the sequencing facility. They should have caught a problem like that. Next, you can actually look at the spatial quality of the sequences on the flow cell. So this is using that same quality information. And once again, across the bottom, you have the position in the read. In this case, this read was 100 base pairs. And across the y-axis, you actually have the position on the flow cell. And it can tell you if there were any problem spots in the flow cell. I've forgotten here to include a heat map, but blue is good and red is bad. So obviously, in this flow cell, in this tile, you have really high qualities. However, sometimes you might see areas where you had an initial drop in quality early, which recovered over time. You also might have a gradual decay in quality, for example, this tile. It's important to note that mappers do take into account the quality of a base pair when mapping. So something like this is just a small loss of tile quality. I would not be very concerned. However, if you have a major loss in flow cell quality, for example, all of these positions had a huge drop in quality throughout the entire read, I would consider going back to the sequencing facility and asking maybe what happened here. Was there a failure of the flow cell? However, a lot of these reads can be saved by just trimming out all the low quality reads, and you have several high quality reads that you can continue with. The last way to assess the quality of your reads is to look at the per sequence quality score. So instead of averaging all the reads together, now you're asking for a single read, what's the average? And you can look at the distribution of that. In this case, along the x-axis, we have the quality score or the Fred score and this is averaged over each entire read. And then on the y-axis, we have number of reads. And so you can see that most reads have a really high quality score of 39 to 40 on average. This represents a really high quality library. This I'm showing is an example from that last picture of the flow cell where you had a drop in quality for several positions on the flow cell. So you can actually see that reflected here in this distribution. You have a small number of reads here that have a very low average quality, and you have a large number, you have an equal number of reads here at that have a medium to low quality score, average quality score of 27 or so, and still a large proportion of reads that have a really high quality score. If this were my library, I might consider trimming all reads that had an average quality less than 30 for downstream mapping. And this can be done with a trimmer, which I will talk about at a later date. So next, you can assess anything that comes before the sequencing by looking at other library qualities. I've split this into two different sections. The first is actually looking at the nucleotide content of each read. So first, we can look at the percent nucleotide representation at each base pair. So this is the sequence content across all bases. Along the x-axis, we have the position in the read. This is the 51 base pair read. And then on the y-axis, you have percent of that nucleotide. In this case, these reads came from human, which is about 40% GC. So in this case, you can see 20% G content across all bases, just about, and 20% C content across all base pairs. And you can say the same with the A and T at 30%. This represents a really good balance. However, you can see some waviness at the beginning of the read, and that can be due to bias. In this case, this is an example of random hexamer bias. If you use random hexamers in your RT reaction in order to produce cDNA, you will see at the very first about 10 base pairs of your read a biasy for different hexamers, which will be represented. And you can also actually see along the read here a little bit of adaptor, which is much more pronounced in this example. Each peak basically represents a nucleotide of the adapter, and you can almost read it off as G, A, T, C, G, G, and so on. This is actually an overrepresentation of adapters that you can computationally remove later. What you really want is for these to be smooth lines and not spiky. The next thing you can look at is the per sequence GC content. So like I mentioned, all organisms have a fairly well-known ratio of GCs to ATs. And in this case, humans, it's about 40% GC, and what you can see is in the red line, which represents my actual sequence, I have about 40% representation of the GC nucleotides. And then the blue line here is a theoretical line. But what happens when you have something in your library that's overrepresented and it's therefore not at 40% GC? In this case, I'm showing an example of an overrepresentation of adapter product. And so the Illumina adapters in this case that I was using had about 50% GC content. So when I have an overrepresentation of those adapters, I have a huge spike at 50%, which is going to skew my distribution. So if I see problems in this distribution, it can mean something, some contaminating sequence. Another example of contaminating sequences could be biologically contaminating a sequence. This spikiness represents overrepresented sequences that have these specific percent of GC content. In this case, it's actually mitochondrial RNA, because this was done in muscle cells, heart cells, that have a lot of mitochondria, and the ribosomal removal did not remove all of the mitochondrial RNA, and so you actually have this spikiness due to mitochondrial RNA. You might also see this if your ribosomal removal did not go well, or if you weren't even doing a ribosomal removal step or a polyase selection step. When Illumina can't properly read a nucleotide, it will instead assign an N to that position. So a large amount of N's at a single position can mean that Illumina had a problem reading the nucleotide at that position or across several positions. I would say that 90% of the time you're always going to see a flat line here and hardly any N's called by Illumina. In this case, I do see it right at 10 base pairs. About 10% of my reads have an N in them, and this was due to a bubble in the sequencer, and so therefore an incorrect read. If this is a really large proportion of your reads, I would consider contacting the sequencing facility. However, mappers do have a method for dealing with N's, so it's not totally detrimental to your mapping, but can affect downstream mapping. Another way to look at the nucleotide content of the read is to look at sequencing length distribution. When you get these from the sequencer, 100% of your reads should be the determined length, 150 base pairs, 50 base pairs, depending on what you're sequencing. However, if you want to look at the FastQC again after trimming, you might see something like this. In this case, the library fragments were smaller than the sequencing reads, and so we removed adapter from the end of these reads, And so we actually got a shorter distribution of some of our reads, but most of our reads were about 125 base pairs where we expected them. But we did trim them, so this is how this can look after trimming. Okay, so other things we can learn about library content as a whole is first, and I think most importantly, something to talk about is duplicated sequence. I think that FastQC is really cool because it actually calculates the percent of sequences remaining if you deduplicated. Here I'm showing an example of a really good library. There's almost no sequence duplication, and what this shows is the blue line is my sequences, my total number of sequences, and the red line is what the sequences would look like after deduplication. How FastQC determines duplication, unlike some other programs, is to actually say, for this specific read, first base pair to last base pair, is the read identical to any other reads? So that's how it's determining this. So here's an example of a library with low complexity. So the first thing I want to point out is on the right, you can see there maybe 8% of the reads have over 8,000 copies of that particular read, or 8% of the reads are actually duplicated. This is likely adapter reads, because it's really the only place you see this. However, if you look at across the board, there's several copies of a lot of the reads, and so this is actually a low complexity library. In this case, this library was chip-seq and over-amplified, so I actually had to remove deduplication after mapping so that I could only look at unique reads. If you do something like RNA-seq, you might see biological sequence duplication. In this case, at some portion of the reads, 10% of the reads are there are about 10,000 copies of single genes, and then a small portion you have even more copies. This peak here is actually due to ribosomal reads. So if you don't properly remove your ribosomal RNA, you're going to get an overrepresentation of reads that come directly from the ribosomal RNA. In this case, it's actually about 50% of the reads that were, or 40% of the reads that were actually lost to ribosomal RNA. So it's really important to optimize your ribosomal RNA removal steps. FastQC looks for overrepresented sequences in large polymers, so the actual 50 base pairs sequences that are overrepresented. And it shows them here where it shows you the sequence that's overrepresented, the number of reads that it found, the percentage of your library, and then any possible source. FastQC comes automatically loaded with the Illumina adapters. But you can, if you know of certain sequences that you're likely to see, such as ribosomal RNA, you can actually also load those sequences into FastQC and it will look for them. However, what I do is I just take the top hit, I take that sequence, and I blast it against whatever organism I'm looking for, in this case, human, and I know that this is ribosomal RNA. So my ribosomal removal step didn't work as well as I wanted it to. So next, you can actually look at the adapter content. This is what we'd expect to see. No adapter content along the x-axis here, which is position in read, and the percent adapter is on the y-axis. However, if you have two small library fragments, in that case, your fragment you had for your library was smaller than the amount that was sequenced, you're going to start reading into the adapter as you move along in the sequence. So right here, at about 70 base pairs, you start reading into the adapter. So this library was a little bit too small for these 125 base pair reads. And so what you can then do is later you can trim off the adapter in order to help with downstream mapping. The last thing that FastQC can calculate is K-mer content. So I mentioned before, it looks for large polymer sequences, but it can also look for smaller K-mers. In this case, it's looking for, it's actually looking for 7 mers. So it can look for anywhere from 5 mers to 7 mers. depending on the setting, the default is to look for 5 mers. You may do this if you have removed all adapter and are still looking for contaminating sequence. However, it will almost always, FastQC will almost always find overrepresented K-mers, and they will often just be adapter that was missed. I wouldn't really worry about the K-mer content unless you are looking to figure out what's wrong with your library under special circumstances. I want to tell you right here that don't worry if your library doesn't look perfect. I don't even really know what perfect means, and just because it doesn't look perfect doesn't mean that it's bad. There are things you can do to alleviate some of the problems I've shown you, such as trim your reads. You can trim off low quality reads or reads into adapters or adapter-adapter dimers. You can also remove duplicates if necessary. I suggest removing duplicates from DNA sequencing, whole genome sequencing, as well as chip sequencing, because with chip sequencing you have such a low input, you're likely to have amplification over amplification. I also want to point out that mapping takes into account base quality. So if you have some quality problems, it is handled somewhat by the mapper, but can be made better by trimming off any poor quality reads. And you can always get more coverage. If you, say, have a lot of contaminating ribosomal RNA, you can always sequence your samples further to get the kind of coverage that you need. Like I mentioned, you can do things like trim to make your libraries look a little bit better. I'm going to cover this in another video, but I wanted to show you an example of how libraries can change after trimming. Here I'm showing an example of adapter dimers in my library. And like I mentioned before, you can see this by this spikiness up and down that actually is adapters contaminating the library. If I trim out those adapters, however, my lines are now really smooth, really beautiful GC content and everything. Like I mentioned, GC content, my adapters are at about 55% GC. So I see a big spike here when I'm looking at GC content. However, if I trim out my adapters, that spike goes away and I'm just left with this little shoulder that I'm not too concerned about. Before I did the trimming, the FastQC determined that I had about 1.5% of my library was adapter, and after trimming, I had no overrepresented sequences. So stay tuned for the next video about how to actually use FastQC.